I'll tell you today about a story that's unpublished and ongoing, and the short title of what Maria Carmen just said is like, how is the gut microbiome of infants with milk protein allergy changes over time? So this is work, again, from my lab at the Hugh University, but also still a collaboration with the Broad and Mass General when I was still a postdoc a few years ago. So the story begins with what is food protein-induced allergic proctocolitis, or we'll call it here FPIAP. So in general, this is a non-IgE-mediated allergy. Okay, so it's not like your sorry, I'll put a timer on so I don't know where I go. So it's not like your sesame or peanut allergy later in life. It starts really early. It starts at around 1 to 4 weeks of age, and you'll see this in our data in a minute. Statistically, if we look at the data, we see that the most common reason for this FPIP is cow's milk, and the next one is soy and rice and so on. And fascinatingly, it self-resolves roughly by 6 months to 1 year for sure. Okay, and so what is how is this really diagnosed, and what is the cohort that we have? So this is what's called the GMAP cohort. It's a cohort that's based in Massachusetts, and there have been about 1,000 children enrolled to this, and about 900 who've passed some criteria of inclusion here. Roughly, if we speak about US overall, we expect to have about 5% of this kind of allergy in early life, and this is like a staggering 17%. So we have 153 infants that are positive for these FPIP, and 750 infants that are controls. So again, this is a prospective cohort, so they were enrolled as they were born and came into the PCP, the regular well visits at the clinic, and then we just followed them and saw which ones developed allergy and which not. And the way that we diagnose this FPIAP, the allergic response, is we see that they're very fussy, and the parents report discomfort, but that is common to many babies. Those of you who are parents in the audience would know for sure. And so what is done here is that there's actually a test for blood in the stool, and if you find blood in the stool, that means that the gut barrier has been breached. And what you do next is you start avoid different kinds of foods, and then you see once when does it improve. And so the first line would be to remove cow's milk protein, and that's indeed the case for the subjects that we're talking about here. So I just want to stress that this is the proteins in cow's milk, so this is whey and casein. It has nothing to do with lactose, which is the sugar intolerance of milk. Okay, so there was a first paper published by Tori Martin, who's the clinician working on this cohort, and the first one was observational. It has nothing to do with the microbiome, and I'll just show one figure from it that states how the initial or the diet over time of the infant has great impact on the chance of developing this milk proteins allergy. Okay, so what you see here is the different infants are grouped into whether this is the initial diet, so really at the first time point they came to the clinic, or if you want to look at the diet over time, and you see that kids that are fed formula, exclusive formula, have a much higher rate of developing the milk proteins allergy. Most interesting is I want to say that early exposure to mixed feeding, that is breastfeeding and cow's milk formula, actually is associated with the lowest rate of FPIP. Okay, so what are we going to talk about today? So we took a subset of these 950 kids, and we're talking about 160 infants here. We chose 81 allergic kids and then chose matched controls for those. So these are the numbers you see here. They were followed for a year, and we have samples at birth, 2 weeks, 1 month, 2 months, 4 months, 6 months, 9 months, and a year. And we have stool samples from all these cases. We also have questionnaires, so we know their diet, their initial diet, and later on at the various time points. And we also know, of course, which I didn't mention, a few more other variables such as, C-section versus vaginal deliveries and others that maybe we'll touch upon in a minute. So this is overall the samples that we have. We have about 960 samples. These are all 16S data. Here on your right, you see that the x-axis is the age of the sample in months, and the y-axis is the different subject. So each horizontal line here would be a different subject. Each dot is a sample, and we've color-coded these samples based on the disease stage that they're at. So all these gray ones are the controls, as you see here. And if you look at kids that developed the allergy at some point, you can divide them to the samples to those that are pre-symptomatic, so prior to any symptoms being reported by the parents. They're symptomatic, which is like the darker reddish points. And then at some point they were diagnosed. So again, that was with blood in their stool. And then you have this light gray bar that would indicate the duration of the allergy, and at the end it resolved. Again, different kids resolved at different times. Some had a much shorter disease period and some had much longer. And following the resolution, we call all these samples in the resolved disease state. Okay, so now we can ask all kinds of questions. We can look at comparisons between, by and large, FPIAP and control, so allergic versus controlled. And we can also look at specific disease stages and try and compare controls to the specific disease stages that we're interested in. We'll start with the first and then we'll get to the second. So the first two big questions that you always see in microbiome-related studies, right, is first is richness. Do we see any kinds of differences between the allergic and the non-allergic? So here you see over time, each set of box plots is by the different time points, and they're color-coded as before by the disease stage. And you see that while there is, of course, an increase in richness over time, as you would expect in all infants, there isn't much of a difference between allergic and control kids in this setup. Okay, so that's the first question. The second question is like, okay, so maybe richness is overall the same, but we would see great differences in abundances of specific compositional changes. And so here again, this is an average plot of all cases, all allergic kids, and all controls. And you can see the ends of how many samples we had in each one of these bins. And again, you see that overall, there aren't that big differences between them. Now, on the one hand, you can say that this is, this is not surprising, right? Because these are really gross changes and we don't expect them to be seen on such a resolution. And so what we do now is we want to dig deeper and try and see eventually what do we still find any differences between these two groups of children. So the next step was to apply a multivariate linear regression model, and we used Maslin 2 from the biobakery from the Huttenhauer lab, and we've controlled for the age. Of course, we've controlled for the subject because we have multiple samples from the same subject. We control for the delivery mode, and we control for the infant diet. And finally, we also controlled for the probiotics, and we'll come back to that in a minute. So if we look at samples from age 0 to 2, so this is the first 2 months, okay, and one of the questions that we wanted to ask is, can we see any signal, at the early time points before perhaps the disease started? And the two most significant results, all results that I'm showing you here are significant, statistically significant, is that we have a higher abundance of Enterobacteriaceae species in the cases and in the allergic kids, and we have a higher abundance of Clostridiales species in the controls. Okay, so that's the first thing that we observed. Next, we said, okay, instead of just clumping it by age, can we also look at the different kinds of disease stages? So we can ask, if we just compare the last pre-symptomatic to the control, so we have all these matched samples, we still see this enrichment in Enterobacteriaceae. If we look at the first symptomatic compared to, again, these matched controls, we see the same kind of signal as we see in the 0.2, which was reassuring. And finally, when we look at the resolved, we see a different story, and that is that we see that there's a higher rate of lactobacillus species in the cases and also a higher rate of Blautia species in the controls. So the first thing to note here is about probiotics. And so when kids are diagnosed with the allergic proctocolitis, the first line of defense, together with avoidance of food, is giving them probiotics. And so we would expect to have more lactobacillus in kids who are allergic just because they were given those probiotics. So now we can actually separate this analysis and look at just within the kids who got no probiotics and the kids who got all the probiotics. Here is just showing the control. And if I add the others, you still see that even within each one of these groups, there's an enrichment in lactobacillus in the resolved compared to the controls or the pre-symptomatic or the symptomatic here. Okay, and so we wanted to take another orthogonal approach to try and see if there are any other kind of taxonomic differences between the two groups, right? So in the multivariate analysis, we kind of examine each taxonomic feature separately, and we wanted to ask if there's something altogether that would separate them. So we took a random forest approach for classification. So just to give a very, very, very quick view into what we're talking about, the idea is that we train a model. We're not going to go into all the details, but basically now, given any new sample, so suppose I'm drawing these balls out of a basket, I need to classify each one of these balls or samples, if it's a control sample, a pre-symptomatic, a symptomatic, or resolved. Okay, so one way to present this result is by a matrix. This is like the true label. So this is what happens when I pull a ball out. I know it's a control, and I want to ask what kind of label do I predict it to be. So this would be the prediction here. And so indeed, 37% of control samples I label as control. And if we continue with the diagonal, which is all the true labels of pre-symptomatic, 49% are being actually labeled pre-symptomatic, and so on and so forth. To resolve, there are 74% of them are being truly classified. And so here's the full picture of all the different categories here. Interestingly, if we look at the features that are most important for this classification, we find some of those previous taxonomic features that we found in the multivariate regression come up here as well, which is nice. And I'll just finish by showing that we also examined what happened at 6 months and onward samples. So right, most of them by then are resolved. And so when we just train a model to separate the control and the resolved, we do a much better job here. And this is really interesting. This again is mostly driven by lactobacilli and other species that we've talked before. It's interesting because kids who have this allergic proctocolitis at early age are more likely to develop the IgE-mediated allergy later on in life. And so we're wondering if there's any reminiscent signal in the microbiome that may play a role later. So to conclude, when we compare these FPIP, these allergic children, and control, we find no large compositional differences between the two groups. We do find significant species that are taxonomic differences between the two groups. Again, mostly Enterobacteriaceae, Clostridiales, and Lactobacillus. If we take a totally different approach of random forest, we find similar differences, and we are able to classify these samples pretty well. And finally, again, there are significant differences following resolution, which is part of what is really interesting for us in this project moving forwards. And with that, I'd like to acknowledge a lot of the people who have done the work, so I'll just go very quickly. We have Victoria Martin, who is the clinician at MGH, who's helped establish this cohort and continue to follows it. Yamini Virkud was working together with Tori. Hera Vlamakis was at the Broad and helped a lot with setting up this project. And Shiana and Wayne have been the initiators of this GMAP cohort altogether, and Eric and Ramnik, my postdoc advisors. And finally, the work was actually done by 2 students in my new lab, Ehud Dahan, who did all the multivariate analysis, and Zvir Itzkovich, who did the machine learning approach. And this is a shameless plug to say that if you're looking for a lab for doing a postdoc, especially if you're computational coming to the microbiome field, please let's be in touch. That's it. Thank you so much.